This is going to demonstrate how to use Cytoscape to create a network. So first go to App, Reactome FIViz, Gene Set Mutation Analysis. And here you choose the latest version, which is 2022 in this case. And now you need to import the genes that you want to create a network with, either by browsing and opening a text file, which should be organized with all genes on separate lines, or by copying and pasting the list. And now you can see that we've imported 548 genes. You then go down and start selecting the parameters. First, you will fetch the functional interaction annotation, and you're given the choice to choose or not whether to include linker genes, which are genes that are not in your list, and that will participate to connecting your genes within the network. I generally always choose this option. This takes a while to create then a network, but the first display of the network is not very useful. It's a ball of string, and it's very difficult to see what's happening. The first thing to do to improve the display is to look at the linker genes, and you can see these appearing with a tick mark in the table for the nodes. And here in the network, they're differentiated from the real genes by very pale green. So here I'm going to first select all the linker genes that have been introduced in the network and make them appear onto the network by clicking Select Node. And now you can see that all the linker genes are highlighted in yellow. Because they're selected, now I can change their style. So I go to Style, Node, and I'm going to change their shape. So here I click on to diamond. You can select another shape if you wish. And now you can see that all the linker genes are now in diamond versus the real genes that are in round. Now, the next thing to improve the display is to change the color of the linkers because now they're black and it makes everything very difficult to read. So I select the whole network and go to change the properties of the edges. So I can also go to select all edges. And now I'm going to the Edge tab and select stroke color. And here I'm given a choice about many different palettes to use to change the color of these edges. So you can go for RGB, or here if you go to HSL, now I'm going to choose, for instance, a gray pale color for the edges, so that now they do not obscure the organization of the network. So then you still end up with a ball of string, which is rather unhelpful. And so we're going to modularize the network, which enable to organize the network based on connectivity between subsets of genes. So you go to cluster network, and now you can see that different clusters have been created, each with different colors, and a new table has been introduced with the list of different clusters with the number of nodes associated to them. And now you can select a module and it will display the module in yellow as selected. This is still not very useful because it's still a ball of string, so we're going to go to layout and change the layout of the network. A good place to start is to go to Group Attributes Layout and now select Module. And now it reorganized the network according to modules with each circle displaying a separate modules based on connectivity. Now we're going to import a table that contains the log fold change ratio into our network. So we go to import table. We click our files, that is a CSV file. And when we click open, we can see that the content of the table is shown. Here, the name have been chosen as the key. We're changing this to the genes because these are the names on the nodes of the network. On the other hand, the name field is not very interesting to us, so we won't import it. But we will keep importing, of course, the log fold changes. So we select this in an attribute, we click OK. And now this table has been imported in, and if we go to the node table, we can see that a new field has been introduced with log2 ratio. And if we go to the nodes of our network rather than the linkers, we can see that we see the values. So now we're going to change the color of the node based on expression level. So we go to the node color field, and we go to the bypass, and now at the moment it's based on nodule with a discrete mapping with different colors per modules. We change, instead of module, we now go to log2 ratio, and instead of a discrete mapping, we're going to have a continuous mapping with colors from blue to red to show a difference in expression. These colors are not intense enough for our taste, so we can go now within the display and change for more pronounced colors. And so we can change the color of every tab accordingly, and if necessary we can also increase the number of tabs, but I'm not going to show you this here. Just going to work with the basics. And once we've selected the colors that we want, we click OK. And now if I go back to network, I can see that the colors of the nodes have been changed. In greens are the linker, and blue to reds are the genes that are part of our network. And we can see that some nodes have got more intense colors than others because their different expression is higher. Now we're going to analyze more in details what these different modules are associated with, and we go to analyze module function, and we have choice between a lot of options, including pathway enrichment. If we click on there, we can see that we have a minimum number of nodes of 3, because that's the smallest nodule, but we can change what we consider as the minimum number that we want to choose. And here we can either include or exclude the linker. Generally always include the linkers because they participated into the making of the network. And now if we go down, we can see that we've got a new table, pathways in modules that have been created, with, for instance, module 0 being shown to be associated with a lot of things to do with actin cytoskeleton and cell migration. And if we go down the table to module 1, we can see that now module 1 is associated with very different pathway terms. And here we can see, for instance, that E1 alpha comes up twice, so that might be something interesting to look at. And again, if we move down now to module 2, we can see that now module 2 is associated with DNA replication and DNA damage. So each module has been really isolated in a way that not only is hyperconnected, but also this hyperconnection corresponds to actual difference in pathway. Now, if we want to look in more details, we go to create a new network. out of a module that we select. So we select from selected nodes with all edges, and now we can see that we've created a new sub network out of our initial network. And we can double click on this entry and give it a name that will remind us what this is all about. So this is module 0. And here this display is not very informative because it's very busy and the nodes are overlaying with each other. So we can again go to layout, and we have a wide variety of layout to choose from. But it is a good place to start to look at the module in organic layout. And if we do that, we can here, for instance, reorganize the nodes the way we want. But here it's interesting to see first that the edges have got different qualities. So either bidirectional arrows like here, or directional arrow like here, or others that linkers that are not are different. And so now if we go to the edge table and select an arrow, we can see what this arrow corresponds to, and we can see that, for instance, the first gene, P4HA2, catalyzes collagen. If we select a bidirectional arrow, we can see that this is a bidirectional relationship with one node activating the other and the other node catalyzing the other. Here we've got an edge with no arrows, and this corresponds to complexes, so protein-protein interaction. And we've got also dashed non-arrow links that are predicted interaction based on database outside of the human interactome. So now we can go for more precise layout, and for instance here we're going for hierarchical layout that enables to lay the nodes in an organized fashion, and this is like an electronic circuit. So if we go and zoom in, you can see that the nodes are organized in a way that maximize the visualization of how they're connected to each other. And so what you can see very quickly is that you've got some nodes that have a very high number of connections, like this one here or this other node here, while in contrast you have other nodes that have very little number of collections to others. And so we want to highlight this because nodes with a high number of connections are known as hub genes that are particularly important in the way the network works. So the first thing that we're going to do is go to Tools, Analyze Network. We're asked whether the graph is directed, but because we don't have only arrows, but also protein-protein interaction, it's non-directed, so we don't click this, and we click OK. And now we've got a small table appearing on the side with analysis of connections within the network, and we've got also sub tabs that we can click in, and this can be exported. And one thing that is analyzed as part of the network is the way nodes are connected together. So we're going to use this to improve the display of the nodes. So we go to node size, and we go to map. We select the column, and here we've got a lot of centrality measurements, which corresponds to the way nodes connect to other nodes in the network. And for instance, we have degree, which corresponds to how many links each node have with other in the network. And here we select degree and continuous mapping. So the default setting makes the node too small to see them properly, so you want to change that. You double click on the minimum size, and a good size to start up with is 50. And the maximum size, you can put any value that you just fit, but 200 can be a good one. And now if you press OK and go back to the network, you can see that nodes with a large number of connections are now much bigger than nodes that have very little number of connections, which are much smaller. And so this enables you to visualize easily hub nodes within your network, which corresponds to nodes that are particularly important for the organization of your network. And therapeutically, this can be very interesting to target, because it means that by targeting these nodes in particular, you will be more prone to impair propagation of the signal within your biological network. So once you've done that, you can also reanalyze for that subnetwork pathway enrichment. And again, you're given the choice whether or not you want to include the linker. And here I include them. And now you can see a more precise analysis of the pathways involved in that particular subnetwork. It gives you the list of genes for each terms, the false discovery rate associated with p-values, as well as measure of how many proteins within your network are part of the signatures that are identified. So now if I go back to the original network, we've done pathway analysis there before. We can also do some further analysis, for instance, for gene ontology terms like biological processes. And here again, I choose to include the linkers. And these can take a bit longer than the pathway analysis to display the results of the analysis. And as an output, when the analysis is finished, you get a new table now being created on the top of the pathway modules. Now you have a Go biological process in module table, again with the terms associated with each modules. So now if you're interested to know about if you're interested in exporting this table, you can right click and click export table and give it a name and save it in a folder, and this will come as a text file that you can open in Excel. You can also export the network as a network file that you can open in other network making software, or you can export as an image that you will be able to put in a PowerPoint presentation, and you've got various formats. SVG is the best format because it will enable you to ungroup the image later on if necessary, and also it's higher resolution. You also should save the session, save the Cytoscape session on a regular basis. Sometimes Cytoscape can crash, and so if it crashes in the middle of your analysis and you haven't saved anything, you will lose all the work that you've done. So I advise you to save on a regular basis. So this now concludes this short presentation on how to build a functional interaction network in Cytoscape. Thanks.